Can we really say it's hot out when it's only 99 degrees? I don't think so. It's not even triple digits. So after I filmed my most recent video about the pygmy rattlesnakes with Jenna Palmisano, I had a ton of extra footage, including what you're about to see. It's literally just 30 minutes of Jenna dissecting snakes in front of me to look for pentastomes and showing me her normal process. Obviously, there's going to be a content warning. We are going to be dissecting snakes. There's going to be blood and guts and gore. It is all scientific. This is very normal stuff. But one of the snakes in particular, in fact, the first one that you're going to see, he's looking a little gruesome, a little worse for wear because he was kind of run over by a car. So without further ado, here we are dissecting snakes. His 30th. Okay, do I need gloves for this, or can I have gloves?
You can have gloves. You don't have to touch anything.
I mean, I'm not gonna not get my hands dirty, so.
He always wants to touch it. So Shasta pulled out a couple of them because she was nervous that the snake was too decomposed, that they would start to liquefy. So she pulled these out.
So this is a roadkill snake.
Yep.
So this looks like a black racer.
Yes, sir.
And what you're pulling out right now are pentastomes that somebody already extracted.
Yes, because she saw them moving around, and she was like, I don't want to lose them. And so
They were like crawling around on the ground?
So they were crawling inside of his viscera. So usually when a snake dies and or gets stressed out, the pentastomes are going to move from the lungs and try to find oxygen elsewhere. So he's a little bit still frozen, but that's okay. We can work with it. So she sliced him open on the side of the road because she was just going to leave him there.
Is this a SLAM member that did this?
Yes. Yeah. So Shasta is one of our great collaborators. She's a citizen scientist. And so she saw this snake on the side of the road and was like, all right, let me just check it. It smells really bad. So she was just going to leave it. But then she found a bunch of pentastomes in it. And so she was like, okay, I'll just take the whole thing for Jenna, and froze it. And so we're looking at this snake a little frozen because he is going to be quite smelly otherwise. But even a snake in this type of condition, we can still detect pentastomes. So these are the ones that she found and pulled out for us. The biggest ones are the females, and they're brown because they have the snake's blood within them. So all that brown and red stuff is actually snake blood that they are digesting or have digested. And so Shasta already did most of the hard work for us by opening this snake, and we can start to see a little bit of the lung tissue here. And so a lot of times the pentastomes, again, will move after death. And they can move down or they can move up into the trachea. And so this is all lung tissue here.
This right here.
Yeah, so it kind of looks like a honeycomb structure. So their lungs are a lot different than ours, a lot more simple. And so the anterior portion of the lung is what's going to be kind of the main respiratory service. And so that's where we see more red. So this is the lung as well. He's a little frozen, so we're just kind of breaking some ribs here. And so,
I don't think he cares.
No. So now we're moving into the trachea. I think Shasta might have done a great job for us and pulled the majority of these pentastomes out. But I just wanted to search to make sure she didn't miss any. And so this is the trachea here. Shasta knows to cut right through it. And we're going to look at a more healthy snake in a second so we can get an idea of where we should be looking. But even a frozen specimen like that, you'll see the pentastomes. They're pretty obvious, as you can see here. But yeah, so that is the lung of the snake.
So when these pentastomes, they live their life cycle within the snake's lung, when they die or molt, because they're going to be shedding their skin and the outsides of their body, where does all of that waste go?
Goes right into the lungs. And so it could be kind of expectorated by the snakes. Snakes don't have diaphragms, so most of the time it's just going to kind of sit there until it fully kind of disintegrate or dissolves.
It rots
within the
inside
lungs.
their lungs.
Yeah. Yeah, so it can cause sepsis and it can cause pneumonia, yep.
You've told me before that black racers like this have the ability to handle pentasomes a lot better.
Yeah.
What is the difference between how a black racer like this handles a pentasome versus something like a pygmy rattlesnake?
So we don't really know. That's one of the things that I want to figure out, if there is some type of molecular component to the susceptibility. We don't really understand that so far. But with black racers, it could also be due to their feeding ecology. So black racers are really good in urban environments and other settings at picking up prey items. So they're eating quite a lot. So maybe that helps them overall not only pick up more of the pentasomes, but maybe that gives them the energy to be able to withstand these infections. I love pygmies, but they're not the best predators, unfortunately. So they're not going to be eating quite as much as a black racer. So they might not have the kind of reserves physiologically to deal with the infection.
So the black racers are going to be eating the cockroaches that are coming from out of our houses.
No, though they're going to be eating the lizards and geckos and things that have eaten the cockroaches and then become infected.
Okay.
Yes.
All right.
Yeah. So I'm thinking that this guy, Shasta, has pulled out all of the pentastomes from him. And so we'll get measurements on those pentastomes. Got a big old heart.
Can you see the heart real quick?
Yeah, so we can look at the other guy's heart, which will be a little bit prettier, but typically they're more strawberry shaped. And this guy is pretty frozen still, but yeah, sometimes you can find pentastomes like down by the stomach as well. But it looks like this guy's she got most of them here.
Can I see? Can you put one of those pentastomes on my glove?
Yeah, you want a big one?
Sure.
Okay.
Just like right across my hand.
Yum. So that's the head.
So this is the head of it. And you can see just how big, and this is probably a female.
Yes, it definitely is a female.
That, you know, in the 6.5 years that she could potentially live is just going to be shedding eggs her whole life. Now, you told me before that a pentastome, they only need to mate once.
Yes.
Right? And they can hold enough sperm to fertilize for the rest of their life.
Yeah. And with relatiolids, they likely don't live as long as 6 years. They probably live a couple of years, but we just don't have the evidence. The only evidence we have of pentastome longevity is in porocephalids, and those do live 6 or more years. But so up here, there will be a spermatheca organ, which is a...
Pull up for 1 second.
You're good. Just smushed her a little bit.
Yeah.
Okay. So up here is going to be the spermatheca.
Yeah, so there'll be a spermatheca up here, and that is where the sperm will be stored, and then she will selectively filter out the sperm and fertilize eggs. And the eggs will kind of fill the entire body cavity.
They don't have any eyes or ears or nothing that we would recognize as a face.
No, they don't. I mean, so pentastomes means 5 mouths. So at one point we thought their hook-like appendages were actually 5 mouths, but there's 4 hooks and 1 main feeding appendage, which is kind of in the center. But yeah.
What's really striking to me about this is that if you cut open a snake without knowing what you were looking at, this would just look like any other part of it. It doesn't look like this is a separate animal from the snake itself. It looks like, I don't know, it looks like a piece of tissue or an organ that would normally be a part of it. I think that we as people who live our lives, hopefully mostly parasite-free, forget how frequently parasites affect things and just how prevalent parasites can be in nature. And that for many animals, that something like this is a fact of life every single day.
Yeah, not all parasites are bad, absolutely. So I think that's an important thing to note. But these ones I do think are bad, at least in naive hosts. Okay, she split apart there, yum. So, I mean, with these ones being this degraded, I'm not going to get an accurate measurement. So I'm just going to put down that they had at least 2 females and what looks like 3 other males. So I'll just mark that down, and I'll take these under the scope later on to get pictures of their hooks, but I will do DNA extractions on them and confirm their identity with a novel PCR assay for relatiolids. All right, 118 liver, cool. So this one we're going to prep for the museum and also take samples for Skylar Hopkins.
Okay.
So for this guy,
species is
Who do you think?
Well, I don't
Who
know.
do you think?
I don't know the genus species of a corn snake.
You-
I grew up calling these red rats. I didn't understand why they were called corn snakes until I was an adult.
You can just put the species code Pagu, so it's Pagu. Pantherophis
Capture
guttatus.
date?
This is from Constantino, so I need to check. Alright, so capture date is 10/3/23.
Location?
So the lat is going to be 28.50386, and the long is -81.27333. Cool. And so you can circle full specimen and liver. Those will be what we take for the museum. And other samples taken, you can write Skylar or just Sky. And so she is studying snake fungal disease in a list of species that she wants me to collect swabs on. And so she sent me swabs where I just go up and down the body twice. And she's interested in snake fungal disease in, again, like 15 different snake species. And so I send these to her. Looks like this animal was actually in shed. You can see a brighter color underneath there. And so we like to be... These snakes aren't alive, and they were frozen, and so we don't know how much of the pathogen might still be on there. So we do kind of a thorough...
You're not afraid to be a little rough with them.
Yeah, rub down, and we'll do the other side as well. And we do this first just so that we're not contaminating, and I've changed my gloves, the specimen that we're taking for her. And if you're ever taking swabs for people that are going to do DNA extraction, it's easier to do the extraction if you chop it off low to the actual tip. Just FYI. Yeah.
For anyone collaborating with Slam, you heard it here.
Yeah, so if you're taking cloacal swabs, please chop it off at close to the tip. So we're going to do SVL, and I'll tell you what it is to write it down. So... Alright, so it's going to be 68.8, and the tail length,
Need a finger?
yeah, on the end of the tail. Let me flatten it for you. Whoop. Alright. Whoop. And the tail length is 12.7. Cool. Alright, so now we can start the dissection.
I like that it has these like pre-described grooves for
Well,
the snake.
so
I don't know if that's
actually,
intentional.
no, I just flipped it so I wouldn't contaminate, and I can just
gotcha.
bleach the whole thing. This is actually supposed to like fit into the grooves of the board below.
It looks like it could totally
We
be
fit
like,
the snake,
oh,
yeah.
I'm gonna fit the snake right in there.
So I just start at the cloaca as a point of entry.
Start from here
Mm-hmm.
and work your way all the way up to the head.
Yeah, so since we are looking for other parasites, I do that, but also the pentostomes move after death, so we're just moving up the body.
Now this is a corn snake. Have pentostomes been detected in corn snakes as well?
Yes, so pentostomes have been detected in Pantherophis guttatus. The prevalence is quite low from what we've looked at, which is kind of consistent with the prevalence that we mostly see in ectotherm eaters, and these guys are mostly going to be eating mammals, but they do eat ectotherms too, I'm sure. So they could be getting infected through either route. I don't know an incredible amount of their ecology or how much they really eat mammals compared to ectotherms, but
I used to have one of these snakes as a pet when I was a kid growing up.
Mhm.
So it's like, I don't know, I have a soft spot for these guys.
yeah,
They're
totally
really,
get
really
it.
cool snakes.
Well, he's got plenty of fat. He was doing good.
So what's the actual fatty tissue that we're seeing?
So the fat itself is going to be all that white stuff.
That right there.
Yep, so that's all fat.
Cool.
And we'll pull that out and measure it. The intestines. Cool. Those are this nice dark spot. Those are the kidneys.
Right here.
Alright, so we'll keep going up. Ugh, snake's shedding. So I kind of just pull up while I cut to try not to get into the organs so that I can kind of look at the anatomy and not just like go right into the body cavity. So yeah, that's more fat there. All that white stuff is fat. Looking at some intestines.
Now you said that this snake was caught in a fence and died from overheating.
At least
This
that's
wasn't-
yeah, that's what we think. And this was also given to me by a SLAM member who also gave me a pygmy rattlesnake, which was infected and I'm assuming died from infection of Relictilla orientalis. But yeah, this was from a middle school actually, so definitely an urban environment. But hopefully we see a nice healthy lung. That's kind of always my goal. It's like, okay, maybe this will be a boring dissection, but I'd rather it be boring than see increasing prevalence. But unfortunately, every dissection day comes with at least one infected snake. So that's the nice pretty lung right there. Thank you. And there's the heart. So the one thing I didn't do is I didn't check the mouth out yet, but we'll do that. Cool. So this is where I just kind of start to spread open.
So we're seeing the trachea right here.
Yep. I cut into it a little bit by accident, so it is open. But then what I do is I usually go down towards the end of the lungs and cut in.
So these are the lungs right here, these dark spots.
That is actually that's the liver.
That's the liver.
Yeah, so.
I'm a marine biologist. I'm not a snake biologist.
That's alright. And so this bubbly portion here is the lung. And there's actually Relictilla orientalis in there. I was not expecting that.
So this is one of our pentasome
This-
parasites.
Oh, I was really not expecting
No.
that. Oh no,
So
that's...
this snake is now infected with the parasite, and it's just sitting right there in the lung itself. You see it right there.
There's actually two of them.
There's two.
So if we... There's
So
one.
that's a little teeny tiny one.
That's a male, probably. And then this is probably a female. Could
How many-
also be a male.
How
But
many corn snakes have you looked at that are infected with your parasite?
out of mine, just through SLAM collection, I don't think any, to be honest with you.
Wow, so this is a first.
At least for my SLAM stuff, yeah. So Melissa Miller has documented it, so it's by no means a novel finding. One of the novel findings that SLAM has contributed to broader knowledge is the mud snake. We found 2 mud snakes infected with the parasite, and that was a new finding. So Ferantia abacura in southwest Florida.
So you saw how the parasites were literally just sitting inside of the lung tissue. They just sit there. So just imagine having one of these, let alone 2 or
100.
100 of them, like some species have, just hanging out and living their lives in your lungs while you're trying to breathe. They're pooping in there, they're breeding in there, they're laying their eggs. Their entire life cycle is happening within your lungs. And there's no way to get them out, because how are you going to get them out of your lungs except for taking an endoscope and sticking it in and literally pulling them out like the most terrifying claw machine ever.
Yeah, so that's a nice healthy heart though.
So, by all accounts, even though this snake was infected with at least 2 pentasomes, even though the snake is infected with at least 2 pentasomes, how would you assess the overall health of this snake based off of what you're seeing?
So there's a couple of SLAM kind of core dissectors, and what we've decided is better than body mass and length of the snake is weighing how much fat they have. And so what we'll do is we'll pull out all of the fat tissue and weigh it and kind of get it in grams of the amount of fat that this animal has. And so that's kind of a better measure because we don't know how long the snake was dead. We don't know if it's dehydrated or something, or if it's kind of been just sitting there for a while and liquefying. And so the mass in relation to its body size might not be the best estimate as to its body condition. So we've started to measure the fat, and maybe that'll give us a more realistic look of the comparison between infected and non-infected snakes. And so I would say that overall this snake has really good body condition given the amount of fat bodies that we're seeing. It was going into a shed cycle, doesn't appear to have other parasites. It looks like it only had 2 reliatielids. So overall this snake looks pretty healthy. I have seen, she actually has some developing follicles. You can see those there.
Follicles for
Babies?
Oh, for eggs?
Yeah.
So this is a female?
Mm-hmm.
So that's what these little bubbles are.
Yeah.
You just point at those.
Yeah.
Again.
So that's what these are.
Mm-hmm.
Right here, follicles. Now, and the reason that we're measuring fat content, the reason that we're measuring the fat content of the snake is because there's some evidence that pentosome infections, they might stop feeding, or they might be get a little skinny from not feeding properly. Is that it? Is that the reason why you're measuring fat content?
Just overall, the impact on the body condition of the host is really what we're looking at. Maybe it has to do with feeding. Maybe it just has to do with, even if they're feeding, they're not able to get a lot of energy. Maybe it's more so impacting other species more than others. So like black racers, for instance, I've cut open ones that have like 65 pentostomes in their lungs.
Wow.
And they'll be yolking up follicles, so they're reproductively doing well, and they'll have plenty of body fat. So we're just seeing kind of different severity and different responses physiologically in different snakes. So yeah, we're just trying to collect as much data as we can to further understand what the risk is to native species and whether, certain species are at a higher risk. So a lot of unknowns.
A lot of unknowns.
A lot of unknowns, but
It's-
hopefully this data will help us understand the disease a little bit more.
Well, that kind of goes back to the whole point of, like, you're kind of on the cutting edge of all of this.
Yeah, it's definitely a frontier of research, for sure. Pentostomes kind of occupy this clade of crustaceans that is relatively understudied and kind of brings a huge knowledge gap to crustacean evolution overall. But now that it's causing concerns in wildlife, people are more apt to study it. But it still is kind of like a big frontier of science for sure. I think this one is the female because that white bit is, I think, the chitinous spermatheca. It's usually the most obvious thing. And so the first one, I think these are probably 2 immature females. The first one is 29 millimeters, and then the second one is 22 millimeters. Cool. The last thing that I'm gonna do, well, not necessarily the last thing, but I'm gonna take a sample of the liver for our museum. And that is a nice healthy liver. I think the liver is like one of the prettier organs in vertebrates. So we'll keep that for the museum. Cool.
I think it's cool that you're saving not just specific organs, but also prepping the entire animal itself for certain museums, for their collections and everything, making sure that as much of the animal is going towards science and future science as possible.
Exactly. That's definitely a part of the goal of SLAM, is just to kind of contribute to broader bases of knowledge. If someone wants to study snakes 100 years from now and they're interested in some Florida snakes and across the southeast, then they can go to these collections and know what kind of parasites they had, what other samples were taken for them, and also get an idea of if morphology has changed over time. Yeah.
So I'm Zak Cole,
I'm Jenna Palmisano.
and thanks for checking out what makes Florida so wild. You did it! I'm going to give a big thank you to Jenna and all of her lab team, as well as my friend David and my partner Rhea for helping me film this. I hope you like this video, and if this is something that you're interested in, leave a like on the video or tell me down in the comments, or do both. I'm trying to put more content out more frequently, so if this is the kind of thing you're interested in seeing, let me know. It really helps me inform on what I'm going to focus on for my next videos. So with that being said, I'm Zak Cole, and I'll catch you on our next adventure. If you want to see the video about pentostomes and pygmy rattlesnakes, be sure to click right here.